So let's take a look at 519 now, the next problem. So with this one, we're continuing from problem 518, and we are trying to determine what would happen if we crossed this female with this male. And instead of trying to determine the distance between SB and CU, the problem tells us that SB and CU are 8.2 centimorgans apart, and wants us to figure out the phenotypic ratio in if we see about 1,000 offspring. So what would be the outcome of the cross, assuming that equal numbers of males and females were observed? So we can ignore the males and females. So again, even if crossing over occurred in the males, it wouldn't matter here because the males are homozygous for stubble, wild type, and curly. So crossing over is only happening in the female. And let's look at the genotypes of the female, the gametes. So we can see SBCU and plus. So these are the non-crossover gametes. And we also have SB and plus for one of the crossover gametes, and plus and CU for the other crossover gamete. Now, because these genes are 8.2 centimorgans apart, if we looked at 1,000 offspring, so 1,000 offspring from this cross are going to come from 1,000 eggs in the female. And because these genes are 8.2 centimorgans across, that means 8.2% of this 1,000 should have resulted from a crossing over between SB and CU. So 8.2% of 1,000, if you break out your calculators, you're going to see that 82 of the 1,000 are going to be the crossover gametes, are going to be progeny resulting from crossover gametes, and 918 are going to be progeny resulting from the non-crossover gametes. And so we're going to assume that these are in equal levels here, equal amounts. So 409 of the eggs should have that genotype. 409 should have that genotype. 41 should be SB plus. And another 41 should be plus CU. Now all of these gametes are going to be fertilized with just one sperm, just plus CU, one sperm with one genotype plus CU. So all we have to do now is interpret the phenotypes of these eggs over here, fertilizing those eggs with these sperm. Okay, so in this case, maybe I'll just draw these over here. So SB CU over plus CU for this one, plus CU for this one, SB plus CU for that, and plus CU plus CU. Okay, and we can put the phenotypes right next to these. I'll draw little squigglies here for fun. SB, so this is going to be stubble because it's dominant, and curly because it's recessive mutant, but these flies have 2 curly alleles, and this is going to be wild type because we have no stubble mutant allele and only one curly mutant allele. Now these flies here are going to be stubble because we have one dominant stubble allele, which is dominant to wild type, and they are going to have normal wings because we have a plus CU allele, right? Because the plus is above the CU, so we know that this is really a CU plus. And I'm not going to even write normal wings. We're just going to write the mutant phenotype stubble. And here, these are going to be curly. And I'm not going to write long hair or anything, but there's no stubble allele here. And here, we're homozygous for the curly mutation, so these are curly. So the answer to the question is we should see 409 stubble and curly, 409 wild type, 41 stubble, and 41 curly. And we should see those numbers because we looked at 1,000 offspring, and the stubble and curly genes are 8.2 centimorgans apart.